folks. Please welcome Morgan from SUNY Oneonta with her research. Thank you.
Thank you everyone for being here this morning, right before break, so we'll try and get through this so you guys can go on break and get your coffee. I'm Morgan Fleming. I just graduated from Oneonta with my master's, and before I dive right into this, I just want to say that as we see changes in fish taxonomy, that's important. It's just as important when we see changes in the parasite taxonomy of the fish. And when bringing the two together, fish and parasites, if we don't know what we're looking at, then we can't really do much more with that. So just to preface this talk, I wanted to put that point out here. So I will be presenting on the second half of my master's. I presented on the first half last year, so we're going to dive right into it. So fish can serve as definitive hosts in some parasitic relationships. This can cause damage to the fish. We don't know exactly how, as I said, unless we know what we're looking at. In some ways, this can also help fish, and ways it can help fish is by sequestering heavy metals. So our parasites can absorb the nutrients, but as they absorb the nutrients, they can also absorb the heavy metals that the fish are dealing with in some of their environments. Some of the parasites, a group of parasites that does this are the Acanthocephalans. They are also known as the thorny-headed worms, and as I go through this talk, I will be referring to their thorny heads as the proboscis. And they are intestinal parasites of your fish. So just to start, there's the two-host life cycle of the Acanthocephalans, where it starts in the definitive host as an adult, and the adults will reproduce, their eggs will go out in the feces, the ostracod will then consume the eggs, and from there, once the ostracod is consumed by the fish again, the adult is now back in the fish. And then, life cycle, it just keeps going. So, I'm specifically looking at the family Neoechinorhynchidae. Neoechinorhynchidae consist of 159 species of 18 genera worldwide. The key features to this family of parasites is the trunk has no spines on the outside of it. There are hypodermal nuclei, they are giant, and there are 5 of them in this organism. I got an arrow pointing to one, but there's a few of them. You can see they're giant. When you're looking at them underneath the microscope, you can't miss them, if I'm being honest. They have a syncytial cement gland, which is right there, and they have relatively few hooks on their proboscis. So, there's been limited studies utilizing DNA sequence data, and in the studies that have been done, there is evidence of the paraphyly of the genus Neoechinorhynchus. So, here are some line drawings of the 18 genera worldwide of Neoechinorhynchidae. Sorry. As you can see, they all look fairly different from one another. This is a world map of all of the Neoechinorhynchid species diversity worldwide, and I am primarily focusing on the North American species, with 46 between the United States and Canada, 10 in Mexico, and 4 in Panama. So, the 10 North American genera. These are our 10 North American genera. We have Neoechinorhynchus, Octospinifer, Gracilocentus, Tanniferephus, Floriocentus, Atactorhynchus, Octospiniforoides, Polycentus, Eocolus, and Myrhynchus. And unfortunately, as some people have pointed out before, there are no common names for these, so it's all scientific names. So, last year I was here presenting on my fieldwork at the type localities to collect these specimens, and just to go back over, these are the type localities for some of the species of the Neoechinorhynchids. I'm just going to briefly show it and then continue because we have more fun things awaiting us. So, the scope was one to collect the samples, which I presented on last year, and for this year, I analyzed phylogeny, and this phylogeny that we put together is the first phylogeny of this family of the North American genera. And then we use the phylogeny to map the morphological characteristics on it. And overall, the objective of this study was to better understand the relationships among the Neoechinorhynchids and look at the patterns of hook evolution. So my methods, the worms were photographed, sexed, and measured prior to sequencing, and the maturity of the females were noted, whether they were gravid or not. And the worms, we vouchered them prior to sequencing, so we cut them in half. Depending on the worm and the size, you might have taken a little more than half of the worm and just saved the proboscis for a voucher that we could deposit into museums for later studies. We did DNA isolation, PCR, sequencing, and then we used 2 markers for nuclear DNA, the ITS and the 28S. Our phylogenetic tree was then generated, maximum likelihood phylogenetic tree was then generated, and we examined it in the context of the morphology of the organisms. So, this is our ITS only tree. And then I will also be referring to the joint ITS and 28S phylogeny. So in these phylogenies, I have clade A, B, and C, and I have them all color-coded to go with it as I talk about each one of them. So instead of starting with clade A and B, I'm going to start with the largest clade that we have here, which is clade C, our blue clade. And this is our clade of the genus Neoechinorhynchus. So these are all Neoechinorhynchus species that we have sequenced. And this phylogeny confirms the paraphyly of Neoechinorhynchus with Octospinifer and Eocolus nesting within that clade. And we have the other genera separate from this, so that's a good thing from this. Then I'm going to jump over to the ITS only tree, in which we have the genus Neoechinorhynchus, still our blue clade. We also have the Mexican Neoechinorhynchus species, which seems to be the sister to clade A. With that, we can say that the Mexican Neoechinorhynchus are not the Neoechinorhynchus genus, with the way they are on this phylogeny. With that being said, we can look at the morphology of Neoechinorhynchus, and it's possible that it is the ancestral form of the family. And when I talk about the ancestral form, I mean the proboscis morphology in terms of where the hooks are arranged on the proboscis in 3 rows of 6 hooks each, or 3 circles of 6 hooks each. Okay, so that's Neoechinorhynchus. From there, we're going to go back up to clade A, where I have Octospiniferoides from the eastern mosquito fish in Florida, and Gracillocentis from the gizzard shad in Illinois. And also, if we were to look back at our ITS only tree, Myorhynchus from various cichlid species is also a part of clade A. So you might look at this and wonder what they all have in common. Well, before I get there, I have to take a step back. I've been talking about hooks on the proboscis and the way they're arranged, but what's the morphology of the hook? It's actually quite simple. There's 2 parts to the hook. We have the blade, which is the part of the hook that is on the outside of the proboscis, and it's protruding from it. And when the parasite infects the fish, this is the part of the parasite that embeds in the intestine of the fish. And then we also have the root, which the root is in the proboscis underneath the skin of the proboscis, the tissue, embedded in the tissue. So with that, the root is going to play a large role in this next part, where what do they have in common? They have markedly large hook roots for each of their rows of hooks. And I do have arrows pointing to them. But don't worry if you can't see, I'm going to go through them also in a little larger scale. So this is Octospiniferoides, and if you look, the hook root is much larger than that blade is. And it's here in context too. So the root and the blade is up top here. And that root usually extends from where the blade protrudes from the proboscis and is also all through there. That's the root. And then there's also Myorhynchus and Gracillocyntus, which I kind of like the way this one looks. It was funny looking when we saw it. Okay, so then that leaves me with clade B, our green clade. So, our green clade consists of 4 genera of 2 families. It's a well-supported clade, as I have here, circled in green, with nodal support of 100. And then its just position is unresolved within where it is in this whole phylogeny, with the nodal support at 76. So this is potentially problematic. Why is this problematic? Well, as I said before, I pointed out those giant nuclei on the dorsal body wall, and I said that Neoechinorhynchids have 5. Parateniocetus, which is the other family, the species from the other family, has 10. So that puts into question the morphology that we use to identify the families. And don't worry, I did do replicates, because when you have something like this, where you're seeing it and it's saying, no, this family member from a different family is within the Neoechinorhynchids, you just don't want it to be true. You want to maybe hope you did something wrong the first time, but no, that was not the case. When I did replicates, they ended up back in there. So it was kind of hard to believe, and then we just did replicates to get a better idea of it. So, in summary, most genera were separate from Neoechinorhynchus. Neoechinorhynchus morphology seems to be the ancestral form. Neoechinorhynchus species from Mexico are not in that genus. And Eocolus and Acanthospinifer are nesting within Neoechinorhynchus. Some genera have morphological patterns that correspond to clades, as we saw with clade A. And other patterns are potentially problematic, calling into question the use of dorsal body wall nuclei for the family, as we saw with clade B. So what needs to happen from here? The membership of Neoechinorhynchus needs to change by adding Acanthospinifer and Eocolus. And then after that, which is far in the future, I will probably not be working on that, but obtain sequences of the Neoechinorhynchid genera worldwide, and obtain sequences from other families in the order to test the morphological characters to see if any other families fall within the Neoechinorhynchids. With that, I'd like to acknowledge my funding from the Helmholtzical Society of Washington's Bernard Freed Student Research Grant, the Theodore Roosevelt Memorial Fund of the American Museum of Natural History, the SUNY Oneonta Research Foundation, the Holloway Scientific Achievement Award, and the D'Antonio Student Travel Fund. Without them, none of this would have been possible. And also, without all the students and collaborators that we've had through this project, without them, collecting these organisms also would not have been possible. So, thank you. And with that, I'll take any questions.